Good morning, I am Professor SENTHIL VAIRAVAN from the Department of Advanced Zoology and Biotechnology, Guru Nanak College, Chennai. So today I am here to discuss a very important topic in genetics which has earned a Nobel prize in the field of medicine and physiology, that is lac-operon. This lac-operon explains the regulation of gene expression in prokaryotes. Lac refers to lactose, which is nothing but a disaccharide made up of glucose and galactose. Operons are the closely related genes which is going to metabolize the lactose molecules. And there are several different types of operon in prokaryotes. One such is the lactose operon, which was first discovered by Jacob, Loaf and Monod, and they have been awarded the Nobel Prize in the year 1965. If you look at the structure of the lac-operon, you have three structural genes, namely lac-Z, lac-Y, lac-A, and that is going to produce three different types of enzymes like beta-galactosidase, permease and trans-acetylase. These structural genes are controlled by regulatory sequences like a promoter gene, where the RNA polymerase will come and bind and initiate the transcription. And also we have an operator gene, lac-O, which is responsible for switch on and switch off mechanism of the structural genes of lac-operon. And other than that, you have a lac-I gene which is going to produce a protein called as repressor protein, which is tetrameric in nature. And this lac-operon structural genes will produce a long chain of mRNA, is called as polycystonic mRNA, where you have many cystron units which is going to produce these three different types of enzymes. And the role of beta-galactosidase is to cleave the lactose into glucose and galactose. The other function of beta-galactosidase is to transfer the lactose into allolactose. Allolactose, which is nothing but the isomer of lactose. And the function of galactose permease is to import lactose into the cell. And the function of trans-acetylase is to modify the toxic galactosides in order to remove from the cell. And there are two conditions. What happens when the lactose is added into the medium, and what happens when the lactose is absent in the medium. In the absence of lactose, these structural genes of lac-operon should be turned off. In order to turn off the genes, we have a protein called as a repressor protein, which will bind with the operator gene. When it binds with the operator gene, and it is going to form a loop called as a DNA looping. So this tetramer repressor protein, which will bind with both the operators like operator 1 and operator 2, which will form a loop so that the RNA polymerase cannot move further downstream, so that the translation will be ceased. When lactose is introduced into the medium, the inducer, the so-called inducer allolactose, will bind to the repressor protein, and then it will be inactive, so that the operator region is made free, and then the RNA polymerase sits on the promoter sequence, and then it will initiate the transcription, and then you will find the synthesis of three enzymes which are responsible for the metabolism of lactose. And when I introduce glucose and lactose into the medium of E. coli, the bacteria will first take up the glucose, and then when the glucose is completely utilized, it will switch on to the lactose metabolism. In case when the glucose levels are very low in the medium, that means the energy levels are very low, so that the ATP molecules are converted into cyclic AMP. So this cyclic AMP will bound with the protein called as catabolite activator protein, and then it will bind in the CRP side, and then it will initiate the high level of transcription. When glucose levels are very high, cyclic AMP is not formed, so that the CRP and CAP compound is not formed, so you will have very less transcription levels. And to summarize, when you have high level of glucose and absence of lactose, you will find there is no gene expression. And when glucose is high, when lactose is present, so this allolactose molecule will act as an inducer to bind with the repressor protein, and you will have a low level of gene expression. When glucose is very low, the CAMP-CRP complex will bind to the cap site, and then when the lactose is absent, there won't be any gene expression. When glucose is low and lactose is present, you will have a high level of gene expression. So this lac-operon explains both positive regulation and negative regulation, and this is a typical example of gene regulation of gene expression in prokaryotes.